We are the Gujarat Technological University. Since its inception over a decade ago, GTU has been empowering young and curating minds to realize their true potential. Over 4 lakh inspired students are enrolled with one of the premier academic universities in India. With more than 450 affiliated colleges in its fold operating across 5 zones of the state, GTU, the International Innovative University, your place to move forward.
Hello students, today we will see about the biochemistry VIVA questions, and some important questions are: what are the chemicals, elements for that form most of the living biochemical matter? And this is the answer. Glucose and fructose are example of single sugars. What are described as a building block of protein? Amino acid. This molecular formula for carbohydrates. Which macromolecule dissolve in lipids? What type of organic substances are proteins? What are the monomers of lipids? Fatty acids. So, which type of following proteins are? Which of the following is? What are used in animals as source of quick energy? Fats that are fatty acid with only single sugar, starches, and cellulose belong to this. Plants like sugarcane and sugar beets store the energy as simple sugars. The biological mechanism is responsible for controlling the activity of. In this stable structure, most of the carbon and compounds peptides bonds. The lipids are used by the body as to perform all secondary structure of the protein, isoelectric point, amino acid alanine, salting in, salting out, local arrangement, which of the following amino acids, which of the following is true of primary and secondary structure, 2 types of beta plate acids, characteristic of the globular protein, uh, which of the following statement is not correct? The alpha helix of the myoglobin, some parts of the protein that have a specific chemical structure. One of the following is not usually a force quaternary structure, protein denaturation, hemoglobin but not myoglobin, hemoglobin which is least likely to affect. If a person breathes in a paper bag, you would expect their blood CO2 quaternary structure of the protein, the action of disturbing the three-dimensional shape, sample questions, negative charge was an isolated point, defined comparatively studies, amino acid found in bonds in protein structure is not considered a pyridine, nucleotides of DNA, hydrogen bonds, chemical bond maintaining, nucleoside is a pyrimidine or purine, electrophoresis here as example. Following is found on RNA but not DNA. Messenger here are some nucleic acid section questions. Function of enzyme, uh, conjugated enzyme, component of a conjugated enzyme, side of an enzyme referred to as, effect of temperature on as enzyme catalyzed reaction. These are the enzymatic questions. Enzyme reactions, statements about enzymes, optimal temperature range for majority of enzyme, allosteric activator, enzyme catalyzed reaction, location on an enzyme, catalyzed reaction by apoenzyme, cofactors, chemical reaction, concentration of rate constants, curves, statements about the enzymes, allosteric enzyme, kinetics questions. These are the several terms. Michaelis-Menten kinetics, competitive inhibitor, Michaelis-Menten equation, non-competitive inhibitor, and Michaelis-Menten enzyme. We mix competitive inhibitors, uh, enzyme at its active site, competitive inhibition, non-competitive inhibition, uncompetitive inhibition, ES complex, competitive inhibitor, enzyme inhibition, Line-Weaver book, non-competitive inhibitors, classical uncompetitive inhibitors, enzyme regulation. In that, there are several questions are there. These are sample questions. Describe allostatic regulation of enzyme activity, protein-protein interaction. These are the sample glycolysis. Sample questions are there. Where does glycolysis occurs? Glyconeogenesis. Where does it happen? These are the sample questions. Glyconeogenesis. Lipid metabolism. Where does it happen? These are the sample questions. Oxidative phosphorylation. Student can pose that slide and we can have an question and answer for that. These are the several questions. Amino acid metabolism. Where does it happen? Nucleic acid metabolism. DNA replication. These are the example of DNA polymerases. Different function of the enzymes in DNA replication. Transcription. Translation. Translation important for the protein synthesis. This is a ribosome assembly, how it look like. What is gene? Gene regulation. cell growth and carcinogenesis, complementary sequence, at least, explain hydrophobic interaction, how are proteins separated by electrophoresis, definition of domain of proteins, function of chaperones in protein folding, explain difference between uh, reversible denaturation and irreversible denaturation, molecules contain positive and negative charge, hemoglobin, list at least three types of RNA molecules. What is DNA melting temperature? Purpose of glucagon? What is the consequence of frame shift mutation? Thank you. This is